last week was the exam, and I was gone on Wednesday. So, I think we're still going to be fine, and we'll be able to still cover all of the topics we need to cover. So let's just revisit basic, really quick here with one slide, basic Mendelian inheritance by looking at one of Mendel's monohybrid crosses, where he crossed two parents of different from a single character. In this case, the classic example was comparing purple-flowered pea plants crossed with white-flowered pea plants. Both were true breeding. You know what that means? That means that they are homozygous and are descendants of exclusively that character. So all the predecessors of this plant, as far as Mendel could tell, were purple-flowered. Likewise, for this one, they were all the ancestors of white-flowered. That way, you kind of know what you're getting here. This shows you the, of course, this is the phenotype, purple and white. This is the genotype. The nomenclature Mendel's using is upper and lowercase letters. We'll see when we get into more complicated genetics. We use different nomenclature, as you'll see. We're always using the same letter. Even though we're talking about two different phenotypes here, of purple and white traits of the character flower color, we use the same letter because we're talking about the same gene, the same locus. It just comes in two different molecular versions, a dominant and a recessive version here. Now, of course, the genotype, we see a pair of alleles here, identical alleles, because these are riding on paired homologous chromosomes. We know that in meiosis we segregate those into gametes, and so it wouldn't matter which one is in a gamete that fertilizes the gamete from this individual. It doesn't matter which one because they're homozygous. We end up with a F1 hybrid, no longer a true breed. We see it's heterozygous, but because of complete dominance, it exclusively expresses this trait. Okay, because of complete dominance, and again, that's a traditional Mendelian character. Mendel then allowed the F1 offspring, these F1 hybrids, to produce a second generation by allowing them to self-fertilize, because again, an individual is both male and produces both male and female gametes. All right. In this case now, when this individual produces sex cells through meiosis, the impaired alleles segregate. You've got a dominant allele. So there's two types of sex cells here in terms of this character. 50% of the sex cells will carry the purple allele, 50% the white allele. And to show all possible outcomes, we use a Punnett squares and handy predictive tool in genetics developed by Reginald Punnett. Along one side, the key to a Punnett square is knowing all possible alleles, and that allows you to generate the appropriate Punnett square, whether it's a 2x2 or a 1x2 or a 4x4, what have you. We see in both cases the alleles of both parents are the same because both parents are of the same genotype. All right, and then we just show all combinations here. We see at the F2 genotype, at the F2, phenotype ratio, we see a 3-to-1 outcome that Mendel kept observing that led to his law of segregation. All right. This 3-to-1 purple, white phenotype ratio, it does mask a 1 to 2-to-1 genotype ratio. One homozygous dominant, 2 to heterozygous individuals that have a dominant phenotype, and one homozygous recessive. We talked about in the previous lecture a long time ago how Mendel was able to confirm that these F1s were truly heterozygous by his test cross. We'll revisit a test cross in chapter 13. Okay, that's when you back cross the F1 high heterozygous individual back to the recessive parent, and the offspring reveal the genotype. Remember that kind of aspect there. Okay. I'm not going to read this hybrid cross just because I don't want to spend too much time there. I made the comment that there are human traits that have a Mendelian pattern, that where a single character is controlled by one gene, for which there are two alleles that show complete dominance. Achondroplasia is one example. There's some other ones there as well. But particularly in human genetics, but in many organisms, genetics inheritance shows more complex patterns. we have to kind of expand Mendel's laws. They're not violated. We don't violate the law of segregation where pairs of alleles segregate in meiosis and are repaired in fertilization. But sometimes we have issues where there's more than two genes involved, or where a gene can have multiple character effects. One gene product can influence another. We can have incomplete dominance or co-dominance where both alleles are expressed. All kinds of complexities, but they don't violate Mendel's basic laws. We looked briefly at incomplete dominance, where the F1 offspring of two true breeds had an intermediate phenotype. In this case, red crossed with white gives you sort of a pink there. But if this were followed the law, the idea of blending, which was a sort of an accepted notion of inheritance in the 19th century, people might have said, well, they look like blending to me. I mix red paint, white paint, I get a pink paint. Blending, there you go. But if you allow these F1s to reproduce, the reappearance of the parental phenotypes throws blending out forever, okay? Because once you blend red and white to pink, every future generation is pink. But the reappearance of the parental phenotypes tells us that blending is not the appropriate explanation. In fact, Mendel's main contribution, why he's considered to be the father of modern genetics, is his prediction that inheritance is a particulate phenomenon, that these particles of information that we call genes, even when they're masked in a heterozygous individual, they still exist and can still influence a phenotype in a subsequent generation. Different nomenclature here. We've got a capital C referring to color with a superscript. That's so that when we see this, it reminds us that if we had big R and little r, we might forget and think of complete dominance, but this reminds us that the heterozygotes have an intermediate phenotype midway between. So that's incomplete dominance. Okay, that was one of the complexities we covered. The second and last one we covered before I ran out of time was codominance. In this case, if you inherit two different alleles, like if you have the phenotype blood type AB, these alleles code for different sugars on the surface of red blood cells that serve as immune markers. If you inherit two different alleles from your parents, you express both. You have both the A and the B sugar on the surface of the red blood cell. Okay? So codominance. I don't know if we saw this slide last time. I think we might have. These show you the phenotypes. Normally we think of a phenotype as like blonde hair, brown hair, but in this case a blood group is a phenotype because it's under the influence of an allele. These are the 1, 2, 3, 4, 5, 6 possible genotypes. We see two alleles, I superscript A and B, capitalized relative to this lowercase I here, so we get an idea we've got some degree of recessiveness and dominance. So there's two genotypes that give you the blood group A phenotype, homozygous for the A allele, or heterozygous for the recessive allele. Essentially, this codes for an enzyme that cannot attach a blood sugar to the surface. It's a mutant. Many recessive alleles actually are mutants. Not always, but many. So both of these give you the blood type A sugars on the surface, shown here as triangles. Two ways to be blood type B. Only one genotype that gives you this phenotype, and here is the example of codominance. Both alleles are expressed. Okay? All right? And of course here, if you have inherit two recessive alleles, the most common blood type in humanity, especially in Asia, for this particular surface marker, there are other surface markers. Your book talks about the MN carbohydrate markers, but for this particular one, neither blood sugar is expressed. Okay? So this is an example of codominance, but it's also an example of multiple alleles. Mendel's pea plants, the characters he studied, had two alleles, purple or white, green seed, yellow seed, shriveled or round seed. Pardon me. In this case, we have three alleles. Now it's three. It's more than two, but it's three. There are some genes that have hundreds of alleles, especially what are called the MHC genes that serve as, again, sort of immune recognition markers on most of your body cells. The MHC genes, the proteins they produce, are responsible for allowing your immune system to recognize your tissues as part of the gang. And when they don't match, that leads to things like organ rejection. So they're important genes with many, many alleles. Now, in terms of multiple alleles, in many cases, these are the response of just a single base pair change. That's all. Okay, these single. So we see here. Here's the dominant allele. We're calling B. Here's the nucleic acid or the nucleotide sequence, a part of that allele. And a single base pair change, it's identical except at this position here we've got, instead of a TA base pair, we've got the GC. This gives you the B1 allele. The B2 allele, we just got a misspelling in a different position, otherwise it's the same as the B. What's often called the wild type allele. Wild type meaning that's the type found in nature or the wild world. Usually the recessive versions are usually some derivation from that, and often that result in a mutant enzyme or protein. And we've got the third allele. These single base pair misspellings, they're known as, I think I've given this one before, are this acronym, they're called SNPs. You heard of SNPs? Did I give you this before or not? No. That's an acronym for single nucleotide polymorphism. Okay? And there was a major effort a few years ago to catalog, part of what interests geneticists, especially human geneticists, is what makes us all genetically different. How much is it, genetic variation is it between us non-sibling, non-identical twins? Okay? How many SNPs make me different from you, or from you? Okay? Is it average? And they've looked at several thousand human genomes now to get an idea of the extent of these single nucleotide polymorphisms. Turns out about one in about every 1,300